Hi everyone, welcome to 154th Stanford MedAI Group Exchange session. This week we have Mansour Abtahi from UCSF joining us to talk about their work on segmentation of periarterial and perivenous capillary-free zones in OCTA. Mansour is an AI ML research scientist at University of California, San Francisco, where he develops multimodal foundation models and deep learning solutions for medical imaging to enhance cancer diagnosis and treatment planning. Before joining UCSF, he was a research assistant professor at University of Illinois Chicago, where he worked on AI-driven analysis of retinal OCTA images for early detection and monitoring of diabetic retinopathy. Mansoor, thank you very much for joining us today. Before we start, do you have any preference on how you would like to take questions? Can we interrupt you in the middle, or do you have designated breaks in your talk?
You can ask questions every time that you want. Yeah, I'm okay with that. Yeah.
Thank you very much. In that case, let's make this session as interactive as possible. And without further ado, let me hand it over to Mansoor.
Thank you, Amara, for the introduction. I'm going to present about the segmentation of the periarterial and perivenous capillary-free zones in the optical coherence tomography angiography images. Let me tell you about myself. I'm a research scientist at UCSF, currently working on the multimodal foundation models using CT and PET images with combination by clinical metadata and radiology reports, specifically for prostate cancer datasets. This foundation model can be used for different downstream tasks. One of them is for segmentation of the metastatic prostate cancer lesions, and another one is for classification of the MTN stage of the prostate cancer. Before that, I was a research assistant professor at the UIC, and I developed several deep learning models such as artery vein segmentation, arterial venous area segmentation, and also capillary-free zone segmentation models. I'm going to discuss about these models in this presentation. So before that, let me give you some clinical background that are related to my studies. You know about the retina. Retina is a layer at the back of the eye responsible for capturing the light and converting it to the neural signals and sending the signals to the brain. So you can see the retina in here. Behind the retina, we have the choroid layer, that is a vascular structure, and behind the choroid we have the sclera layer, that is a tough layer responsible for the structure of the eyeball. The retina itself is a neurovascular structure. It has different type of the neural cells, and all of the neural cells are sending the signals to the brain using the optic nerve. The vessels in the retina are originating from the optic nerve and sending all of and spreading through the retina to supply the neural cells in different regions of the retina. We have a macular region in the retina that is the center of the retina, responsible for the sharp vision and high detailed and colorful vision that we have. At the center of the macula, we have the fovea region. As you can see, the fovea region is thinner than other areas of the retina. And behind the retina, we have the choroid and the sclera that you can see in this image. This image shows different layers of the retina. We have different type of the neural cells in the retina that everyone has different duties, but the most important cells are photoreceptors, that has two types, rods and cones. Rods are responsible for detection of the intensity of the light, and it gives us a black and white vision. But cones can detect different colors. We have several cone types that can detect different colors such as green, blue, and red. And all of them are getting the light that is coming from the outside and transfer and converting the light to the neural signals, and the neural signals can be transferred to other neurons in the upper layers, and after some pre-processing in these layers, the signals are transferred toward the optic nerve, and it's going toward the brain. And all of these neurons need nutrients and also oxygen. The vessels in the retina are responsible for supplying these nutrients and oxygens. However, they have choroid behind the retina that can feed some nutrients and oxygen to outer retina, but for the upper layers of the retina, we have some vessels in different layers, in the superficial layer, intermediate, and deep layer that supply the nutrients and oxygen. This image shows a 3D image of different layers of the vasculature architecture. We have superficial capillary plexus, middle capillary plexus or intermediate capillary plexus, and also deep capillary plexus that are shown here. At the superficial layer, we have large vessels, but in the deeper and middle layers, we only have the capillaries. And also, we have different imaging modalities that can help us to take images of the retina and can be used for the disease detection. The classical one is fundus photography, that is a microscope designed for taking the images of the retina. As you can see, it gives us a 2D image that can show the structure of the retina, and it is a wide field of view. It shows the optic disc here, that is the blind spot, and also the macular region and the fovea at the center of the macula. It shows us the structure of the large vessels, but actually it is a 2D image, and it cannot give us a good information about different layers of the retina. But we have optical coherence tomography, or OCT, that was invented in 1991. It can give us information about different layers of the retina. It uses the law coherence interferometry principle that can detect different layers of the samples and gives information at every point. And by scanning across the retina, we can collect information. Every line that you can see here is a B scan. By doing several B scans beside each other, we can collect information about the retina at different locations, and we can construct a 3D image of the retina that in this B scan you can see the fovea zone that is thinner than the other areas, and you can see different layers of the retina, and this is the Bruch membrane, and behind that is the choroid layer. So OCT gives us good information about different layers of the retina, and but for processing the vessels, we need another technique. Optical coherence tomography angiography can give us a 3D map on the vessels. The idea behind the optical coherence tomography is that by scanning several times at the same location, we can see that in some areas, the variations in the signal is very, very low and the signal is very constant. But in some areas that we have flow, there are a lot of fluctuation in the signal. And by calculating the variation of the signals in different areas, we can generate a 3D map that is actually the location of the vessels in every area that we capture the image. And this image shows the vessels at the optical disc. These are the vessels that originate from the optic nerve.
Mansoor, can I stop you right here and
Sure.
if you can go back? So what do you mean by variation? Is it like spatial variation, like as you're
Standard,
scanning?
yeah, standard deviation of this signal that you can see
Oh,
here in that different scans at the same location at every point the intensity or have a variation. We can calculate the standard deviation of these variations that it actually shows, for example, because mostly in the vessels we have the red blood cells,
mhm.
and sometimes there is a red blood cell in the area, but the other time it doesn't exist. Because of this, we have some variations in the signal, and it shows that there is a flow in that area.
Okay, makes sense. Thank you.
Yeah, this image shows the comparison between different image modalities. This is the fundus photography. It has a wide field of view, but it gives us a 2D image. When we are zooming in the fovea, in the macular region, you can see the fovea, and you can see the structure of the large vessels, but not too much detail, and the contrast of the vessels is very low. And also for the OCT, we have same case. This is a end-phase OCT that actually comes from a 3D OCT, but by analyzing the B scans, we can have information from different layers of the OCT. But in here, you can see the OCTA image that shows all of the vessels with high detail, with a high contrast. And this OCTA is for the superficial layer of the retina. And this OCTA is for the deeper layer of the retina. As you can see in the OCTA superficial layer, we have large vessels, but in the deeper layer, we don't have large vessels. We have mainly the capillaries that I discussed earlier. And this image shows the OCT and OCTA images, and you can see differences between them and the pattern of them in different layers. This is the end phase and in different locations, and this is the B scans of the OCT and OCTA. And this is the architecture of the OCTA images in different layers. And this is the OCT. So another concept is diabetic retinopathy. Diabetic retinopathy is a disease that affects vessels of the retina. It is a complication of the diabetes when diabetes is not well managed and we have high blood glucose or high blood sugar level most of the time. It can damage their vessels, especially the capillaries, and it can cause different problems on the retinal vessels, such as microaneurysm, such as cotton wool spots, heart exudates, and also hemorrhages that are blood leakage. And also in the late stages of the disease, we have abnormal growth of the blood vessels. And actually, diabetic retinopathy is a dangerous disease that can lead to the vision loss at the late stages. And this image shows the different stages of the diabetic retinopathy. We have a healthy fundus photography here, but in the mild stage, we have localized swelling of the small blood vessels that in the retina that we call them microaneurysm that mostly happen in the capillaries and arterioles mostly. In the moderate NPDR, they have mild NPDR plus some small bleeds that are called dot and blot hemorrhages and leaks that are called hard exudates or closure that are called Cattonwood spots of small blood vessels. But in the severe NPDR stage, that is non-proliferative diabetic retinopathy, we have moderate NPDR plus further damage to the blood vessels. We have intraretinal hemorrhages, venous bleeding, intraretinal microvascular abnormalities. And we have proliferative diabetic retinopathy that happens when we have new vascular formation or new vessel formations, or vitreous paroretinal hemorrhages or tractional retinal detachment. Also, we have diabetic macular edema that can develop, that is another disease, but can develop at any stage in the mild or moderate or severe. This image also shows different stages of the diabetic retinopathy by the OCTA. As you can see, this is a normal case, and it shows the superficial and deep plexus, and mild NPDR and moderate NPDR, severe NPDR, and proliferative diabetic retinopathy. As you can see, we have some non-perfusion areas in the different areas of the proliferative diabetic retinopathy and also severe stage of the NPDR, and also some areas, non-perfusion areas in the moderate stage. But when you are seeing the mild NPDR, the differences between the mild and normal cases is very subtle, and because of this, we need some image processing techniques or deep learning models to be able to detect differences between the mild and normal, to be able to detect diabetic retinopathy in the earliest stage. And that is very important to prevent prognosis to other stages. For this, we had this idea to separate the arteries and veins to be able to analyze them separately. The main idea is that diabetic retinopathy generally affects the arteries and veins differently, and it is important to separate the arteries and veins to extract quantitative features from them and separately to be able to analyze them. For this purpose, we decided to use deep learning for the segmentation. We developed we generated several ground truth images for the data set that we had. We had a data set on 3 millimeter by 3 millimeter images. For the ground truth generation, we had several criteria to detect which one is artery and which one is vein. One of the main criteria is the capillary-free zone around the arteries and around the veins. As you can see in this image, there are some hollow areas adjacent to the vessels. And as you can see, for this vessel, the capillary-free zones areas are larger than this capillary-free zones area adjacent to this vessel, because of this is the main feature of the artery that has larger capillary-free zones, and this can be used as a good feature that can be helpful to detect which one is artery and which one is vein. That shows that this is artery and this is vein. Another feature is that arteries do not cross their arms, and I mean arteries do not cross arteries, and also veins do not cross veins, and also we can trace vessels if we know that, for example, this is a vein. We can trace to detect other vessels as veins or arteries. And also generally we have arteries and veins alternating pattern in the images, as you can see, because we need arteries to supply the nutrients and oxygen and veins to drain the waste products and the carbon dioxide. They are mostly alternating in the different locations. For example, this is an artery, this is a vein, this is an artery, this is a vein, and this pattern can be seen in the this image. We also developed guarantees for 3 by 3 data set. In the 3 by 3 data set, we have higher resolution, and we can see smaller vessels and also the capillaries. As you can see, capillaries around the fovea zone. And we can also segment capillaries as arterial side capillaries or venous side capillaries that can be used for the processing. The main architecture that we used is AVNet. AVNet is actually a dense net architecture, and we used several multimodal fusion options. One of them, because we have OCT and OCTA images, we can define several multimodal fusion options. One of them is OCT only architecture that use only OCTA images, and the other one is OCTA only. And the early fusion of the OCT and OCTA, and also the late fusion of the OCT and OCTA, that after going through the AVNet, the output can be concatenated, and then we can use a convolution operation and extract the final AV map. We have a data set for the 6 by 6 data images. As you can see, the output for the OCT only architecture, it can detect which one is artery and which one is vein, but when we are comparing with the ground truth, it cannot depict all of the details in the image. But when we are using models that have OCTA as input, we can see higher details in the prediction. And when we are comparing them, we can see that OCTA only early fusion and also the late fusion architectures have competitive performance. However, for the late fusion, we have a slightly better performance compared to OCTA only and early fusion.
Answer one quick question about if you just go back one slide to the architecture.
Yeah.
Just one more, please. Yeah. Here,
Yes.
so when you have these different blocks, this dense block, transition block, and decoder block. I'm just curious, what's the strategy to evaluate? Like, do you add one block at a time and do ablation, or how do you know what is the right combination of blocks?
In this study, we didn't generally design the architecture of the model.
I see.
We use a dense net architecture to do the process. We were mainly focusing on analyzing the fusion studies, fusion options, and see which one is more useful. Because we have tested before that and saw that dense net architecture has a better performance comparing to other models that we have used before.
Okay, great. And one more follow-up is, like, are there like multiple OCT manufacturers, or like, do you have different manufacturers producing different models, and like, can you train on one manufacturer and then test on another manufacturer?
Yeah, that's a very good question. Actually, we have different type of the OCT or OCTA machines that the output of their models is somehow different. However, there is some similarities, but the intensities and some of them are doing some post-processing before giving the OCTA. And in this data set, we used only OptoView machine. However, if we wanted to have a device agnostic model, we should develop a model that has different data sets from different devices to be able to have a consistent performance on all of them.
Okay, thank you.
Thank you. And we also had a 3 by 3 data set that we used that to develop a 3 millimeter model. In this model, we used the same pattern, we used the same multimodal fusion options, and we saw that the OCT only architecture generally can depict the type of some of the large vessels, but not very small capillaries. However, when we are combining the OCTA and as an input to the models, we can have higher accuracy in segmentation of the arteries and veins, and also the capillaries, and also the arterial side of the capillaries or venous side of the capillaries. And also the model's performance is showing that OCTA only early fusion and late fusion has competitive performance. However, late fusion here has a slightly higher performance than the OCTA only, and some yellow rectangles here shows in which areas late fusion had better performance compared to other models. Another model that we developed was about arterial and venous area segmentation. The main idea is that if we...
Mansour, if I can stop you before you move on to your next model, what's the baseline here? Like before your model, what were people using to differentiate between the two?
For the segmentation of the arteries and veins?
Yeah, yes.
There were some models. One of them was UNet that we had before developed in our lab, but the performance of the model wasn't so good. It was good, it can detect the arteries and veins, but the details of the some small arterioles or capillaries wasn't clear. We used a higher accuracy, a more accurate data set in this model, and also we developed a data set for the 3 by 3 data set. And the idea that we used multimodal fusion options, it shows that what is the weight of different modalities and how we can use them. Generally, after doing this process, we understand that OCTA only can be sufficient if we have a limited processing power, because when we are doing late fusion, we need 2 branches, and the model architecture is heavier, and the training process can take longer. So the output of this study gives us this idea that when we are using OCTA only, we are not losing too much accuracy compared to the late fusion architecture.
Thank you for explaining that. I think we have one question from the audience. Winston, you want to go ahead and ask your question?
Yeah. Can you hear me now?
Yes, we can.
Yes.
Okay. Thank you for your introduction. I am interested in the ground truth rules. Just like in previous slides, you mentioned 4 rules, like 2 kinds of the vein cannot cross. No, the same kinds of vein cannot cross each other, right? Yes. Here.
Yes.
How do you apply those ground truth segmentation, or how do you apply those rules?
Yeah, this criteria are generally for the manual annotation process that we are using to annotate the ground truths before doing the training process. During the training process, we are using IO loss function to generate a AV map that is matched with the guarantors.
Okay. Okay. Thank you.
Thank you.
Sorry, Mansour, probably I missed your first part of the talk. So how did you create the ground truth for this task?
Yeah, this is the guarantors generation process is a time-consuming process.
Right.
I use some image processing techniques to detect
Okay.
detect the vessels, but we also use visual inspection to detect which one is artery and which one is vein, and also we had some manual modification at the end to be sure that the guarantors is very accurate. Because we have a limited data set, the accuracy of the guarantors is very good, very important to have a good performance in the prediction. And actually, I believe that if we have a limited data set, the only option that you have is to have a very good,
Very clean ground truth. Yeah, that's
yeah, very clean guarantors to have a good performance.
So somebody manually checked it, you said, right?
Yes, yes, we
So
did
did you also have a like a kind of like a balance between the known diabetic retinopathy and the normal ground truth, or it's like only collected on a particular population?
Yeah, we had this data set, we had 68
Okay.
control subjects and 36 DRIs, but during the training, we oversampled the DR1s to have a better performance on these cohorts.
Got it. Because it's a very, very time-consuming task. So that's why I asked, like, how did you create the ground truth? Yeah.
Yeah,
Thank
this
you.
is the most important and time-consuming part of the project. Yeah. And yeah, also for the 3x3 images, we had same performance, and it can be used for the 2x3 images. I was talking about the AVA-net, artery and vein area segmentation. In this project, we use k-nearest neighbors to classify all of the background pixels as arterial area or venous area based on the fact that it is closer to an artery or it is closer to a vein. After doing this, we have this arterial and venous area map or AVA map. If we multiply this with the OCTA, we have OCTA AV maps that is very useful because we have all of the intensity informations of the OCTA image, but for the arteries and veins separately. In this case, we can extract many quantitative features from this image for arteries and veins separately. And for this, we developed a deep learning model, AVA-net, using this training dataset, and also we tested on this dataset to do quantitative analysis. The architecture that we used here is a UNet architecture, but the main difference is that we use dilated convolution operations because we have this idea that by using dilation, we can get more information from the non-local areas and have a higher performance. And actually, it has a higher performance than the usual UNet architecture. And this is the performance of the model on the accuracy, the DICE score, and the IOU for prediction of the AVA maps. By multiplying these by the OCTA, we can generate OCTA AV maps. This is the guarantors, and this is the prediction. As you can see in yellow rectangles, there are some differences between them, but the overall performance is good, and it can be used for the image processing. Using the OCTA AV maps, we can extract some quantitative features. One of them is perfusion intensity density that can be used for the arterial vessels and also for the venous vessels, and also can be done for the total vessels. By extracting these features, we can also calculate the arterial venous PID ratio that can be shown here that when we are comparing them for different cohorts, we have the control that are healthy subjects and no DR that are diabetes patients without any kind of diabetic retinopathy, but we also have mild DRs that are at the earliest stage of the diabetic retinopathy. As you can see, the PID will decrease for the total vessels between no DR and mild, but there is not a significant change between no DR, but there is significant change between control and mild. And also arterial PID has same pattern, but for the no DR, there is increase between the control and no DR for the venous PID. When we are calculating the arterial and venous PID ratio, that is division of arterial PID by venous PID, they have a biomarker that is significant for all of the groups between the control no DR, and mild, and control and mild. And this is a good biomarker for early detection of the diabetic retinopathy. In addition to this study, we did some other studies. We showed that by doing differential artery vein analysis, we can improve the accuracy of the diabetic retinopathy classification, and also we can predict the outcome of the anti-VEGF treatment for the diabetic macular edema. And also we developed an open source toolbox for the artery vein segmentation that is publicly available, and you can use it. You can download it and use it for the segmentation of the arteries and veins. But let's go and talk about the capillary-free zone segmentation. I talked about the capillary-free zones and what are them, but generally the capillary-free zones are non-vascular areas adjacent to the arteries and veins. As you can see, for the arteries, these are larger than for the veins. The main idea why we focus on the capillary-free zone is that when we have any kind of disease, these capillary-free zones are changing, especially in the earliest stage, because any kind of damage to the capillaries, to the vessels, and the small vessels can show them in the capillary-free zones and the size of the capillary-free zone. We developed ground truces for the capillary-free zones. As you can see, arteries are shown with red, and periarterial capillary-free zones are shown with the yellow color, veins with cyan, and perivenous capillary-free zones with magenta, and the background is black. And this is the ground truth for one sample. We also noticed that the capillary-free zones are smaller for the nasal half compared to temporal half. Because of this, we decided to have an analysis on the nasal half and temporal half separately to see if they can give us a sensitive biomarker. This
Mansour,
is the
just can I ask a background question here? So, is there a limit on like how much of the area surrounding the artery or vein can be considered capillary-free? Because there seems to be like, I mean, then what is background? If you're talking about all of the capillary-free region, then what's the background here? The black pixels.
That's a good question. We have the arteries and the arterioles, and that are actually can be seen here as large vessels. All of the areas that are adjacent to these areas can be considered as capillary-free zones. We cannot extend the non-vascular, non-perfusion areas other than these because it cannot give us a good information. Generally, for example, these are capillaries in this area, and these are capillaries. But we only consider these areas that are adjacent to the large vessels.
Okay, thank you. And I think we have one question from the audience too. Winston, do you want to go ahead with?
Yes.
Hello. Okay. So a very small quick question, like in the subfigure D, if we have some cross section of red vein and the arteries and vein, how do you decide which one is on the top?
You mean in this cross section here?
Yeah.
Yeah. We can see in the P scans, not in the in-face images, and we use some B scans to see which one is at the top. Also, we have some general knowledge that most of the time arteries are above the veins, but generally we should check different B scans to be sure that this is artery crossing over or the vein is crossing over.
Mansour, is it because it's a 3D scan? You should have like complete knowledge of what is crossing the other, like what's over and what's under.
Yeah.
Oh, okay. Thank you.
Thank you very much.
And this is the data set that we used for the training and also for the test. And this is some samples for the ground truth images that we developed. We had healthy control and also NPDR, mild NPDR samples in the training process. I tested several segmentation models to check which one has higher performance. I also developed some customized models, but generally some famous models had higher performance, and because of this, I brought them to the paper. That there are 4 of them are here, 2 of them are convolutional neural networks, and 2 of them are vision transfer models. The first one is UNet architecture. The backbone of the UNet architecture that had the highest performance is EfficientNet V7. The UNet architecture is most of you know about the UNet architecture, and it has an encoder and decoder and some escape connections. And the main feature of the EfficientNet V7 is that it has a compound scaling method that can scale in depth and in the width and also the resolution of the image. It gives us give it a good capability to be able to detect all of the features in the model, in the images, and be useful for the segmentation. And the other model is UNet++. UNet++ is similar to UNet, but it has some additional escape connections, nested escape connections in the process. Then the knowledge is going to transfer it from the encoder to the decoder. We have some dense connections here, and it is an interesting architecture that can learn from different layers of the model at different levels and can have a good understanding about the features and their connections to be able to depict all of the details and have a good segmentation performance. The other model that I use is TransUNet. TransUNet also has a UNet architecture, but the difference is that it has a backbone with the that it's combining CNNs and vision transformers. At first, we have 3 CNN layers, and then we have vision transformer architecture. It has 12 transformer layers that it has multi-head self-attention module that process the key, query, and value. Then it's giving it to the multi-layer perceptron layer. And we have 12 of these modules in the vision transformer model. After that, we are extracting the hidden features and reshaping that from the patch embedding to the image embedding. And we are using convolution operations and upsampling and concatenation with the escape connections that we are receiving from CNNs, having upsamples to the higher resolution to give us the original image size and do the prediction of the segmentation. The other model that I use is Segformer. Segformer is also a vision transformer model. It is also a UNet architecture. It has 4 transformer blocks that each one has an efficient self-attention module and also a mixed feed-forward network with overlap patch merging during the processing. And all of the information from these blocks are transferred to the multi-layer perceptron, and that is processing all of them and upsample and gives is to the next and concatenate all of them together and gives us to multi-layer perceptron to give us the original size. After doing the segmentation and training of them, we saw that the UNet++ architecture had the highest performance compared to others with 92.75% accuracy. After that, we had UNet architecture and trans-UNet, and also after that, Segformer. As you can see here in this problem, vision transformer models didn't have good performance, and I think the main reason is that in this segmentation problem, we had a lot of small details, small capillary-free zones in different areas that most of the time UNet++ architectures or UNet architectures that are convolutional operations has better performance in detection of small local differences. But vision transformers models are very good in understanding of the relationship between different regions of the image because they are patching the image to different patches and have the self attention module to understand the relationship between different area of the image. They had a good performance, but compared to the CNNs, they couldn't outperform them because of this specific reason. And this is the results of the test data set. We predicted the capillary-free zone segmentation for all of the test data set that we had, and we extracted several quantitative features from the capillary-free zone maps. The first one is the ratio between the capillary-free zone area and the corresponding vessels. For example, for the arterial ratio, we have the ratio between the periarterial capillary-free zone and the arterial vessels area, and also for the perivenous ones, the perivenous capillary-free zone area divided by venous vessels area. And we also had the total capillary-free zone area divided by the total vasculature area. As you can see, for different groups, for the control, nodular, and mild, we have an increase in the size or the ratio of the capillary-free zones compared to the vessels. And specifically for the control and nodular, we have a significant change, and also between control and mild. In some cases, in nasal half, for example, we have significant change between the mild and nodular, as you can see here. But when we are assessing the prevenous capillary-free zones, we can see that for all of them, the changes are significant, even for the nodular and mild. in the whole image, in the temporal half of the image, and the nasal half of the image. And this gives us this understanding that perivenous capillary-free zones are more sensitive to the periarterial capillary-free zones. And it can be logical because veins are after the arteries, and any kind of problem in the vasculature structure can show their effects in the perivenous capillary-free zones, because they are, if there is any problem, any kind of leakage, any kind of problems during the process, can show them in the venous side. Also, we did connected component analysis of the capillary-free zones. We compared the number of the capillary-free zones in the images for total capillary-free zones, for the periarterial ones and perivenous ones in the whole image, and temporal and nasal halves. It can show us there are significant changes between the control and nodular for all of them, but there is no significant change between nodular and mild. That shows that this model can detect differences between the nodular and mild, and it can show us even when a patient has diabetes but hasn't developed diabetic retinopathy, there are some changes in their retinal neuronal vessels that are not detectable easily. Also, we have mean area of the capillary-free zones that after the connected component analysis, we calculated the mean area of the capillary-free zones for the total capillary-free zones and also for the periarterial and perivenous ones. It gives us a good understanding about all of the changes that are happening in the capillary-free zones. As you can see, for all of them, the capillary-free zone, the mean size of the capillary-free zone are increasing, and the changes are significant between different groups for the control, nodular, and mild. And specifically between the nodular and mild, the changes are very significant. And for all of the areas, as I discussed, you can see here that the value of the temporal half capillary-free zone sizes is higher than the nasal half. And there are some differences between them that combination of all of these information together can be helpful for a solid classification algorithm that can use all of these quantitative features as an input. We can use traditional machine learning algorithms to do a good classification.
So, Mansour, can I ask you one quick thing here? Because you're talking about all this quantitative feature, right? I completely agree with you. That shows the statistical significant difference between like these three groups that you are targeting. But we still see that there are a lot of overlaps, right? If you look at the 95% confidence interval, you see that there are still a lot of overlaps between these features.
Yeah.
So your primary target, I think your last comment kind of clarifies it. So your primary target is to use these quantitative features and feed it to another classification model, right?
Yeah, generally, these models shows, this statistical analysis shows that these biomarkers are promising, can be used for the classification. If the differences between them wasn't significant, so we cannot use them for the classification. But when we are seeing significant change, this is promising, we can collect all of these information, give it to some traditional machine learning algorithms such as support vector machine, decision trees, XGBoost, and different
So
algorithms.
why do you think that extracting these quantitative features are useful rather than using the OCTA and OCTA images full together?
Yeah, that's a very good question. The main point is here that when we are using deep learning models for the classification, if we wanted to have a solid performance, we should have a large data set. And
Got it.
collecting a large data set, especially in the healthcare, is complicated because there are many exclusion criteria, many problems, and that cannot give us a large data set. But when we are developing some deep learning models that are extracting features, then we are using them for the classification. It is more useful. The other point is that most of the time, the classification models that are done on the imaging itself is not explainable. When we are saying that, oh, it has mild DR, we cannot convince clinicians that it actually has mild DR. But when we are extracting quantitative features, we can show that you can see that here that the mean capillary-free zone size increased, and it is not a normal capillary-free zone size for the healthy subjects or no DR subjects. So it gives us good information about the current situation of the patient, and also we can analyze can do longitudinal analysis when we extract quantitative features for a patient, for example, for 3 months ago, and compare that with current image that we are taking. We can compare them to see the situation is better or worse. And if any kind of treatment, if any kind of prevention we have there, we can understand the result of them, the effect of them on the quantitative features.
Got it. Thank you.
Thank you, Mansour. Just one sort of small procedural question. How are you normalizing these areas? Is it per slice, per patient, this NA, NV, NT?
These are per image. Normalizing the size of the images, you mean? Size of the capillary zones, you mean?
Yes. What's your denominator here? I'm trying to understand that. Have you like cropped the images or to that everything is kind of uniform, or do you need to take into account the size of the retinal region as well?
Yeah.
I mean, is it varying across different samples? Do you need to take it into account, or is it you have already done the pre-processing so that everything fits into the same number of pixels?
Yeah, I got your point. Because all of the samples are 6x6 images, we don't need to normalize them. And also we are calculating the capillary-free zone sizes in case of the number of the pixels, because the size of every pixel is consistent through the data set. We're not going to be worried about this. But if we had different sizes, because some images have different resolutions, we should convert the pixels to millimeter square or some area units. And also for every image, we are evaluating every image as an independent sample, and we are evaluating and measuring that quantitative feature on that image. And also for the temporal half and nasal half, we are considering only those halves.
And in your equations 4, 5, and 6, what is the NA, N subscript A and subscript V?
Yeah,
NT?
the N is the number of the capillary-free zones, because we are calculating the mean size of the capillary-free zones. We have several capillary-free zones, and we are calculating the mean value of mean size of them. Yeah.
Okay. So that's that makes sense.
Thank you. And in summary, we can say that we developed deep learning models that can accurately segment all of the periarterial and perivenous capillary zones in the OCTA images, and also capillary zone features offers new biomarkers for microvascular changes beyond standard vessels and perfusion density metrics. We had some standard density metrics that gives us some information about the different states, but generally they were not able to distinguish completely between the no DR and mild DR because as I discussed earlier, the differences are very subtle. And differentiating periarterial and perivenous capillary-free zones, as well as nasal and temporal halves, can provide more sensitive biomarkers than when we are using traditional machine learning algorithms. It can show how much effective they are actually when we are measuring the classification accuracy. And also results indicate that the potential of all of these features for the early detection of the diabetic retinopathy, that is very important to prevent the progression of this disease to moderate and severe stages. And for the future works, we can develop models, as I discussed, on larger data sets and also multi-center data sets, multi-device, because as I discussed, they have different type of the devices that there are subtle changes between their processing when they are calculating the OCTA images. And also we can contain longitudinal OCTA data set and to have a better model for processing different type of the images. A more generalist model can be useful for this. And we can also extend this capillary-free zone analysis to other retinal and systematic vascular diseases such as diabetic macular edema and also retinal vein occlusion, retinal artery occlusion, and also hypertensive retinopathy and also sickle cell retinopathy. And we can also explore correlation between the capillary-free zone alternation and treatment outcomes that I discussed previously. We showed that we can use artery vein analysis to predict the treatment outcomes of the diabetic macular edema with higher accuracy. And we can also use capillary-free zone analysis to do the same thing. Possibly it can improve the accuracy of the treatment prediction. That's all. Thank you for your attention, and I'm ready to answer any questions if you have.
Thank you very much, Mansour. Thank you for this excellent presentation. I'm sure our audience appreciate all of the background knowledge that you provided on retinal imaging. So before we move on to questions, I'd like our audience to give our speaker a virtual round of applause. And then the floor is open for questions if anybody has any. I know we are kind of already over time, so if you have any further questions, maybe we can put you in touch with the author later. But if you want to ask anything now, please go ahead. Okay, I think Mansoor, since we are already over time, so our audience are kind of dropping already. So if somebody gets back to us, I can probably put you in touch with them. With you.
Yeah,
Thank you for this excellent presentation.
It was an excellent talk, Mansour.
Yes,
Thank
a
you
lot
so
of
much,
background
David.
knowledge. Yeah, I told you, like most of our audience, they don't know much about OCTA, so it was good to learn.
Thank you, David.
Thank you very much.
Yeah. All right. Take care.